Once we can distinguish which is RNA and which is DNA, so with respect to the composition of their bases and sugar, now we can go for how we are going to get that any sugar moiety can get combined with the any base to form nucleoside. So here we can start with what is meant by nucleoside. So nucleoside is nothing but it is one of the unit which is formed by the attachment of a base to first position of sugar. So nucleoside is formed by the attachment of base. So that base means either it will be considered as an adenine, guanine or cytosine, uracil. So that base will get attached to the sugar moiety at which position? That is at first position. Here I have mentioned the position of these carbon atoms present in sugar as 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. So whereas that carbon atoms which are present in base will be considered as C1, C2, C3, C4, C5. In order to distinguish the carbon atoms between base and sugar moiety, just I have distinguished it as 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. So now once any sugar moiety, either it will be ribose sugar, beta D ribose sugar or beta D oxyribose sugar, they can get attached with the base at which position? First position. Hence they can result in the formation of the nucleoside. So here we can guess. So there are two types of nucleosides. So first type is ribonucleoside, another one is deoxyribonucleoside. So ribonucleoside can be formed in the RNA, whereas deoxyribonucleoside can be found in the DNA. Now once you go for how we are going to get the formation of any nucleoside, one example we can consider here. So beta D ribose, when it combines with adenine, what is that resultant nucleoside named as? That is adenosine. The name of the nucleoside obtained by the combination of beta D ribose with adenine is called as adenosine. In case, in spite of combining beta D ribose, if it is beta D to beta D to deoxyribose, then if it is combining with adenine, then in spite of writing the name adenosine, we can write it as deoxy, prefix deoxyadenosine. That is the main difference with respect to the name of nucleosides with respect to RNA and with respect to DNA. And here we can take a list of sugar which is having the combination with different bases to give respective nucleosides. In examination they can give question like this: name the sugar and base present in adenosine. Then you should be able to identify. So in case they have given name the sugar present in sugar or base and base present in adenosine. So they have given the name adenosine. So adenosine is obtained by the combination of, there is no prefix term deoxy, hence which is the sugar you can easily guess, that sugar will be ribose sugar. And which is that according to this name, which is that base we can easily recognize, that is adenine. So ribose combines with adenine to give adenosine. But instead of ribose, if we take deoxyribose, name the sugar and base present in deoxyadenosine, then what is that answer? So deoxyribose sugar, and which is that base? Adenine. So only thing is here we can make the difference. There is no deoxy here, but there is prefix deoxyadenosine. And in case of guanosine, so which is that, as the name itself indicates, that base will be guanine. And there is no deoxy, hence which is that sugar? Ribose. And name the sugar and base present in cytidine. So which is that sugar? So sugar will be, no deoxy means it is ribose sugar itself, and which is that base? Cyt means we can just easily recognize it is cytosine. So it is named as cytidine. For guanine, guanosine. For ribose with cytosine, cytidine. And by the combination of ribose sugar with uracil, it is called as uridine. So in case if it is deoxyribose, deoxyuridine. In case if it is deoxyribose, it is deoxycytidine. And if you go for deoxyribose with thymine, so it is resulting in the formation of deoxythymidine. But if there is no deoxy, only ribose sugar with thymine combination, it would have called as thymidine. So that is how we can just recognize the particular sugar and base which are present in the different nucleosides. So based on the name, we can recognize which is specific sugar and which is specific base. And of course, in the formation of nucleic acid, that is polymer chain, for the existence of polymer chains, we need monomers. So which is that monomer? Here we can consider that is nucleotide, we can guess. So here, once you will get the combination of sugar with the base, you will get nucleoside. Then finally that nucleoside will get combination with phosphoric acid. So we are going to link phosphoric acid group at which position? That is at 5th position, 5th prime carbon position of the sugar moiety. So which is that phosphoric acid group? We can just write it as H3PO4. I can mention central phosphorus group, then double bonded O, here OH and OH and here OH. Now you can just guess here. So to the phosphorus there are attachment of 3 hydroxyl group, right? So now there is a interaction between this OH group and this OH group. Now we are going to remove off water from this combination of CH2OH with the POH group. There is removal of water. With the loss of water molecule, you are going to get the link of this phosphorus along with oxygen to the 5th carbon atom, which is resulting in the formation of nucleotide. Hence, by the combination of phosphoric acid with the nucleoside at 5th position, you are going to get the resultant product. Which is that resultant product? That is named as nucleotide. So here if you take that specific example, when adenosine, so adenosine is combination of ribose sugar and adenine. So sugar will be ribose and base will be adenine there. And when it is combines with phosphoric acid group, then that resultant nucleotide will be named as adenosine monophosphate. Of course, here you can find due to the cleavage of bond between O and H, you will get negative charge here. And here also there is a cleavage of bond between O and H, you will get negative charge on the here. So you will get that phosphate moiety which is get attached to the 5th carbon atom, resulting in the formation of nucleotide. That is how you are going to get the combination of sugar moiety with base, and sugar moiety at 5th position, it is get attached to the phosphate group. Hence, it results in the formation of nucleotide. So, further, these nucleotides will undergo combination with another nucleotide with the formation of phosphodiester bonds to form dinucleotides, trinucleotides and polynucleotides, which are nothing but which are resulting in the formation of nucleic acids such as DNA and RNA. Once you understood what is the nucleoside and what is the nucleotide, so in nucleoside there will be combination of sugar and base, then nucleoside, and along with the base it combines with phosphoric acid to give nucleotide. So such nucleotides will undergo combination, undergo linear combination. So they will just combine with another nucleotide unit with phosphodiester linkage. Then the result in the formation of polymer chain, which are nothing but polynucleotides. So these polynucleotides are nothing but nucleic acids. So finally you are going to get that structure of polynucleotides which are obtained by the special type of bond called as phosphodiester bond. So now we are going to learn that how we are going to get that bond between two sugar units of two nucleotide units. Now let us take the formation of dinucleotide. Here I have given the reaction where you can consider you are going to get the formation of dinucleotide. I am not considering a polynucleotide. Initially you should know how we are going to get the linkage between two nucleotides. So we can consider here dinucleotide. So here I have taken two nucleotide units. As you know, there will be presence of base, sugar, and the one is phosphoric acid group. Here also another nucleotide, base, sugar and phosphoric acid group. Now, once you can guess H3PO4 once once it gets interacting with the fifth carbon atom, that is CH2OH group, then OH from the CH2OH group get interacting with the hydrogen of the phosphate, that is phosphoric acid group, and it will be removed off in the form of water to give this phosphor oxygen linkage to the fifth carbon atom. Now let us consider the same combination of phosphoric acid group to the fifth carbon atom. Now these two nucleotides will interact with one another. How that interaction is taking place? Now you observe it keenly. Here to the fifth carbon atom we have attached this phosphate group, but here because of the cleavage you are going to get this negative charge here. So phosphate moiety you are going to get, and here let me consider this is that OH group will be there. Now this OH group of phosphor getting interacting with the OH group of the third carbon atom. Now we are getting that interaction of the phosphate group of the fifth carbon atom with the hydroxyl group of the third carbon atom. Now we can easily guess OH group will be interacting with a H to give off water. There will be loss of water molecule here, and because of that loss of water molecule, here you are going to get the combination of this phosphor along with the oxygen towards the third carbon atom, So here there will be loss of, to the attachment of sugar there is loss of one water molecule, and this side also there is loss of one water molecule. Two hydroxyl groups will be involved, hence it results in the formation of phosphodiester bond. So this typical type of bond will be considered as phosphodiester bond. This phosphodiester bond is actually lying between 5' and 5' and 3' carbon atom, that is between fifth carbon atom and the third carbon atom. This phosphodiester bond is obtained. This phosphodiester linkage is the main cause for the formation of polymer chain of nucleotides. So here I have given simple sequence of representing the structure of nucleic acids or nucleic acids. Now they are nothing but polynucleotides. Here we can go for sugar. Here we can consider this sugar and this is base. And here we can guess that phosphate group will be get attached to the sugar of the next nucleotide. Then to this sugar there is attachment of base here, and this is also linked to the another phosphate group of it. So now there will be continuity from one sugar moiety to the another sugar moiety. There is a presence of phosphoric acid group that results in the formation of phosphate, that is phosphodiester linkage. Like this you are going to get the sequence of arrangement of the nucleotides one after the another through phosphodiester bond. Hence in between I have mentioned this is n number of such units which is containing sugar, phosphate along with the base. So this is nothing but polynucleotide. Structural sequence of polynucleotide, which is nothing but nucleic acids. Got it? So that is regarding the formation of nucleoside and formation of nucleotide, and finally you are going to get the information, that is we got the information regarding formation of polynucleotides. Once you know there will be definite sequence of arrangement of different bases in DNA or RNA, that is nothing but nucleic acids, then you should know regarding one rule which is called as Chargoffs Rule. So actually this Chargoffs Rule was given by the scientist by name E. Chargoff. So what this Chargoff Rule says? So according to this rule, the main base composition in DNA, so that will be varied from one species to the other. So that means each and every species will have their own arrangement of the sequence of the different bases. So here we will consider that base composition in DNA varies from one species to other species. But what he found that in all those cases, that amount of adenine was equal to that of thymine. So if there are 20 number of adenine, then there will be exactly 20 number of thymine. And similarly, he also found that the number of cytosine was equal to that of the guanine. So, number of adenine will be equal to thymine and number of guanine will be equal to cytosine. So, it was just given, this information was exactly given by whom? The scientist by name Chargoff. Hence, that rule is named as Chargoff Rule. What does this rule state? According to Chargoff Rule, based on the composition in DNA, which is varied from one species to the other, but in all cases, the amount of adenine was equal to that of thymine. Similarly, and that of cytosine was equal to that of guanine. So, it was given by Chargoff. And now, if you go for the structure for these nucleic acids, usually you can consider primary structure and secondary structure for nucleic acid. So, actually the primary structure of nucleic acids will exactly give the information regarding the sequence of nucleotides in the chain of nucleic acid. If you consider chain of nucleic acid, you will get definite sequence of different bases, as you know. And if you go for secondary structure, so there is a possibility for those polynucleotides to wound around each other. That means DNA has got a double-stranded helix structure. So, you are going to get double-handed helix structure like this. That is possible due to the wounding of the polynucleotides around each other. So, now as you can consider here, I have mentioned here, guanine is in combination, hydrogen bond is formed between guanine and cytosine. And you can consider that adenine is in combination with thymine, thymine with adenine, and similarly guanine with cytosine. That means you have that combination AT and here it is GC. So, that structure will be considered as double-stranded helix structure. Hence, what is that secondary structure for DNA is called as double-stranded helix structure. But if you go for the secondary structure of RNA, usually RNA will be existing as a single-handed structure. But very rarely, the same polynucleotide will be wounding around itself to form, again, rarely you can find it, that is rarely they can form the double helix structure that will be formed only by the same polynucleotide chain which will be wounding around itself. And now if you can consider, so other than DNA, if you go for RNA, actually RNA is of three types. So which are those? Messenger RNA, ribosomal RNA, then transfer RNA. So mRNA, rRNA, then tRNA. All these three types of RNAs are much helpful for the synthesis of proteins. So DNA actually it is very much important. It will give the information for the, it will be having, it is acting as reserve for the code of protein synthesis. That means it will send the message for RNA to synthesize particular protein. So RNA as well as DNA plays a major role for the synthesis of protein. So information will be coded in DNA and that code will be decoded with the help of protein and ultimately results in the formation of specific type of proteins. Therefore, both DNA as well as RNA, they are playing a very major role in the synthesis of protein. And of course, now if we consider one of the scientists, the great scientist called as Hargobind Khorana, they won Nobel Prize. They have actually they shared that Nobel Prize along with other two scientists named as Marshall Nierenberg and another scientist, that is Robert Halley. So now actually why they won the Nobel Prize? Because they were able to crack the genetic code, which is that genetic code which is able to distinguish from one species to the another species. And of course, if you go for another important thing regarding DNA, like thumb impression, what we can say, we will be having our own individual fingerprints. Each and every individual will have their own fingerprints. We can distinguish individuals that is by considering their fingerprints. But of course, we can change the fingerprints by operation. But we can't change the fingerprints of DNA. Then what is meant by DNA fingerprints? It is giving the information regarding the sequence of the bases in the nucleotide chains. Got it? That is, it is giving DNA fingerprinting is giving us information about the sequence of bases in the chain of nucleic acids. Actually, what are the uses of this DNA fingerprinting? So, with the help of this DNA fingerprinting, it is very much helpful in forensic lab, that is for identifying any criminals. And even if you go for determination of any paternity, that is we can identify the parent of any particular individual. So, that is nothing but it is helping for determining paternity of an individual. So, that child or any person belongs to whom, we can recognize the parent of that particular person. And even if you go for another important thing, so it is able to identify the dead bodies. How they are able to identify the dead bodies? That is just by comparing the DNA of the dead body with the parents or the children. So, and finally, if you go for importance of nucleic acids, so that is DNA as well as RNA. So, both are helpful, very much helpful for the protein synthesis. And of course, this in case of DNA, it is helping for replication. So, replication is nothing but mitosis, that is cell division. So, one DNA will undergo it will convert into duplicate form of 2. So, it will give two couple of daughter DNA. So, which will be having the same Xerox copy of that parent DNA. So, that is how it is able to give that daughter cells which are obtained due to the mitosis will be having same DNA which is as that of the parent DNA. So, that means this DNA is very much helpful in replication, whereas the RNA as well as DNA, they are very much helpful for the protein synthesis. So, that's all regarding the nucleic acids.